Today we're happy to welcome Christine Parent from University of Idaho, who will be this week's IB seminar speaker. Christine got her bachelor's degree at McGill University in Montreal, got her master's degree at Carleton University in Ottawa, studying conservation ecology, focusing on introduced wasps in the Galapagos Islands. She then went on to get her PhD at Simon Fraser University, working with Bernie Crespi on endemic snails of the Galapagos Islands, a topic we're going to hear a lot about today. She then made a trip to one of the best universities in the U.S., University of Texas in Austin. Worked with Dan Bolnick, and actually we almost completely overlapped at UT Austin. And in that position, she did some experimental evolution work on flower beetles. She then had a short research position at UC Berkeley, and just 2 years ago got a faculty position at University of Idaho, where she has returned to this endemic snails of the Galapagos system. And so that's the subject that she's going to present to us today. So please join me in welcoming Christine.
Okay, thanks for the invitation. I had a great time today meeting a lot of people with very different research interests. And clearly you guys have a dynamic research group, and it's very fun to be here and have the opportunity to present some of my research that I've been conducting in the last few years. Okay, so as Eli pointed out, what I'm going to talk about today is diversity, and specifically diversity on islands. And so I'm going to start with a classic diversity slide. So this is probably or like some version of this is probably the most used slides for biology or organismal biology talks. And yeah, of course it is. What better way to justify all the energy we put into our research and all the money we put in our research than showing how diverse and complex the world is out there. But if you start and take a closer look, then you might start finding that, yes, the world is diverse, but there's definitely repeated patterns. And so when we look at form, function, and structures, we see repeated patterns across very wide ranges of organismal groups. And so this can be, like, as I was mentioning, forms, but also it can include function and, for example, these guys here that find their resources using echolocation, but completely independently evolve mechanism to do that. So I would argue that, yes, the world is diverse out there, but we definitely see patterns where we find over and over again the same type of form and function. And on the other hand, there's stuff that we don't see. Like in theory, you could think that maybe there should be something like that out there, but we don't find it. So the fact that when we look at the diversity out there, we see these repeated patterns and these gaps in the morphological and phenotypic variation out there, to me is indication that behind these patterns, there are some processes, non-random processes that are creating this repeatability and these gaps in the diversity. And so this is basically what I am interested in. I'm interested in understanding what are the drivers of this diversity and what are the constraints. Because if there's gaps in repeated patterns, it's because likely there are constraints in what can be achieved in terms of form and function. And obviously, this is a very broad question. And one possible approach to answer such a broad question is to try to simplify things a bit. And the way I've done that in my research program is to focus on island systems. Island systems, just so that we're all on the same page, can be like these volcanic islands, for example, in the middle of the ocean. But I also consider islands, for example, a set of lakes in the landscape, for example, or mountain tops can also be considered islands. So basically any series of habitat patches that are isolated from one another by a matrix in the landscape that is unsuitable for the organisms. And it can also include islands that you might want to create in your lab. And so I've done also a lot of work, experimental work, as Eli pointed out, while I was at UT Austin on experimental evolution, and my current work also includes some of that type of work, but for the sake of time, I won't talk about this today. Okay, so islands are simple or simpler naturally. So that's one big reason why they're a good system to work on. When you have a series of them, when you have replicates, then you can actually look for repeated patterns across a landscape. And that provides you some information about what processes might be generating these patterns. And then if you're lucky and the islands you're working with are actually part of an archipelago and you have some information about the timing of the formation of these islands, then what you got is a time machine. Because basically each one of these islands provides you a snapshot in time of the process of diversification. And so as you go from a very young island to an older island, what you can see is a time series of the process of diversification and community assembly. And so what I'm going to do today is talk about this process of diversification in the context of island systems. And I'm going to take a closer look at a few components of this diversification process. Namely, I'm going to focus on phenotypic variation in the form of morphology, and then ask the question how this morphology, this diversity of morphology that we see out there, might be the result of the ecological context where these species are evolving, and how physiology of these species might actually constrain what is possible morphologically in diversification. And to do this, we're going to take a little trip to South America. And so we're going to go to Galapagos. So here's South America. And Galapagos Islands is this group of islands that are located about 1,000 kilometers off the coast of Ecuador here, that's located here. So very well isolated, which means that you have a group of islands that have been colonized over time, but because they're so isolated, it's represented by a subsample, or the organisms there are a subsample of all the diversity that you have around the islands. So much simpler to study. So here's a closer look. So I was talking about snapshot in time. So the Galapagos, we know actually quite a bit about their formation. And so they're a hotspot archipelago, which means that you have a hotspot under the plate, and as the plate is moving, you have the formation of volcanoes in a sequence. And so that means that as the plate is moving in this direction, the hotspot is stationary under the plate, and that generates a volcanic activity that's forming different islands in a series. So here's the same archipelago. Now I'm color coding these islands by how old they are. And I'm going to use this coding throughout the talk. So the Galapagos, the islands that we know of today, the oldest ones are about 4 million years old and located here, so on the cooler end of this gradient. And the hotspot is actually somewhere here. And so these are very young islands here. From the perspective of small organisms, organisms that don't disperse really well on land, this big island here, which is huge, is actually composing more than half of the land area of Galapagos. It's actually made of 6 different volcanoes. So if I quickly go back, you can see here the different volcanoes. There's one here, 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 and here. And these volcanoes are actually isolated from one another by barren lava flows. And so there's no vegetation there mostly. And so for a lot of invertebrates, for example, or some mammals and so on, actually crossing this bridge here is as difficult as crossing this water here. So for the purpose of this talk, what I'm going to do is consider these different volcanoes as separate islands. Okay, so let's actually get there. So here we are, just about to land to one of the islands. And so the first thing that is striking about Galapagos is that the habitat is super dry. And on most islands, what you have is a coast that's composed of these rocky cliffs or sandy beaches. And then if you look at the rest of the landscape, it's very dry and arid. So let's get a bit closer here. So now those are the rocky cliffs. So either you have that to climb if you want to get to the island, or you might land on these sandy beaches where you have all sorts of animals that are greeting you. Then if you walk up a bit, really quickly, you start getting vegetation. And that vegetation, as I was saying, is super arid. So it's desert-like. You have these big Opuntia cacti, these shrubby, thorny things that are just really dry pretty much all year round. But if you keep going up in elevation, on some islands, that's all you're going to find. It's like this shrubby, dry habitat. And so for the great majority of the low islands, that's all you're going to find. So mainly the smaller ones. Now, if you start hiking a bit up on the larger islands, then you're going to reach a zone that looks like this. And that's called the humid zone. And it's much more lush and tropical-like. However, in terms of diversity, it's still very poorly represented compared to the mainland. So some of these trees, for example, there are actually daisies. And so in terms of diversity, it's reduced, and the species that have made it there have adapted to fill different niches that were unoccupied. So this is where you're going to find a lot of the famous species. So for example, the finches and the tortoises. Both lineages also occur in the arid zone, but you're going to see them more active and moving around in the humid zone. Okay, so let's keep going up. And so like after probably a day of hiking, eventually you're going to reach on the very high islands an area where you don't get trees anymore. So it's this grassy sedge zone. And so again, very different habitat, much cooler, lower precipitation, and much more open in habitat. So there's a lot of diversity in terms of the potential habitats for organisms to adapt to on Galapagos. And if I take you out and we take the whole day to hike up like this, there's a good chance that you're not going to see one of the critter that I actually study. And this is because you actually have to get down on your knees to see them and start flipping rocks, looking under barks, and searching for them. And so if you do, that's what you're going to find. And so the group I study is a group of land snails. All species are from the genus Nesiotis. The genus itself is not endemic to Galapagos, but all the species are. And as you can see here, they're really diverse in terms of their color. So they vary from being bright white to this photo actually is not a good representation. They get dark black basically, and everything in between. And it's mostly on that spectrum, between white, brown, going to black. So there's no purple snail or that sort of color. There's also a lot of variation in their shape and their sizes. So if you look, for example, this guy here is very slender. It's a very elongated shell. And there's another one that's kind of like this here, up here. And then some of them are a bit more conical-like, and some are almost round, like a sphere in terms of a shape. So there's a lot of diversity on Galapagos in this group. And so when I started studying this group, this was one of my main interests, just trying to understand what happened on Galapagos to generate all this diversity. So to kind of sum up a bit, or give you a bit more details about this group, so I said genus Nesiotis. There's over 80 species that have been described. And every time we go, my research team we find new things that we eventually will need to describe and name. All the species I was saying are endemic to Galapagos, and they're actually all, or pretty much all, endemic to a single island. And the very great majority are actually restricted to a very small distribution range. And as I was pointing out, they vary a lot in their morphology, but also in their ecology. So on that hike up that we just did, you would find them everywhere if you start looking, except on the beaches and not on the cliffs, the rocky cliffs. But as soon as you get like about 10 meters in elevation, you're going to start finding them even if it's super dry and arid. And you find them all the way through the humid zone and the grassy zone at the very top. So why is that so impressive, I guess, or remarkable in terms of diversity? Well, so if you take the whole family of the same group, so the family Butylimulidae from South America, and you start out looking at the shape, the size, and the color of the snails there, and you start out looking at their ecology, they're practically all the same. We're talking about hundreds of species that are distributed all across South America, Central America, all to Mexico, through Mexico and Baja California. All these species have this conical shape. This aperture here, the opening of the shell changes a bit, but they all have that typical cone shape, and they're really smooth, like their surface is really smooth, and their color is also very similar. So what happened when they got to Galapagos? Galapagos is a tiny little set of islands, and when they colonize Galapagos, after a few million years, you have all this diversity. And so really this is what I'm still interested in understanding that process. So if we're going to start thinking about these questions, it's actually going to be useful to think as a snail for a second. And so some of you study snails here, so it shouldn't be too hard. So one main component of a snail, of a snail's life, is its shell, right? So when you look at a snail, that's probably the most obvious part. And when we look at all this diversity, a lot of that diversity can be characterized as variation in this shell morphology. And so let's think about a bit like how a shell is actually formed. And so a shell, simply put, is a cone. So when the snail is born, at least in this group of snails, it has like 1.5 whorl. So when it comes out of the little egg. And then it proceeds in adding to the shell material via its mantle in constructing and building the shell. And really what it is, it's a cone that's twisting around a central axis. And what creates all this variation that we see, whether you're skinny, you're flat, or you're more round, is basically the rate at which this cone is twisting around this central axis. And so there's many ways that this twisting can happen, and this will lead to different morphologies in terms of shells. Now, if we think a bit about the costs and benefits of different shell shape and the sizes and so on, we can try to come up with hypotheses as why we'd see like different shapes and colors and sizes in this group. So because the snail is actually producing the shell itself, we can assume or presume, I guess, that there's a cost to it. And so if you were to build a house for yourself, and there's a cost to the material to actually build that house, you would probably want to maximize the volume of your house for the surface area of your house, right? And so if you're doing that, and if you're a snail, what you'd create is a shell shape that would be the closest to a sphere. So like a very round, looking shape. If in contrast you build something that's really long, that's way more costly, because for the same volume, you need much more material to build that shell. Not only that, if you're long, presumably, in terms of biomechanics, you're going to be probably easier, your shell is going to be probably easier to break than like something that's closer to a sphere. So clearly there's benefits in being round and like drawback of being elongated. So that raises the question, why would you ever build something that's elongated? And so one possible reason for that is that, again, thinking as a snail, snails don't have skin as we do. And so moisture loss is a very big deal for them. And so one way to reduce this moisture loss is to keep your contact with the external world as small as possible. So that aperture here. And if you're twisting your cone around this axis, one way to keep that opening small is to build a shell that's very skinny. And so that's one hypothesis that's been out in the literature for a little while to explain why we would find this variation in shell shape. And so I was really interested in testing that in this group of land snails. OK, so this is what I'm going to do here, basically thinking about like the water loss, so physiological aspect of the snail, and see how that relates to the morphology. And so when I started working on this question, I wanted to know both if this variation across species, if there's a relationship between the environment where you find these species and their morphology, both like across species and within species. And so what I did is I took X-rays of these shells so that I could easily quantify their shape. So I use a geometric morphometric approach to do that. And so I'm not going to go over the details, but like the point is, like, I've measured, now we've added to these 30 species. We have now data for over 65 species in this group. And we are using a phylogeny to control for the fact that these species are related to one another, and therefore they might be looking like similar just because they have their shared ancestry. And at the intraspecific level, what I did is it was actually pretty challenging to find a species that had enough of a wide enough distribution to actually have some ecological variation in terms of like the habitat where they're found. But there is one such species on Santa Cruz Island here, where the species distribution actually overlaps between the very arid zone and the humid zone. So now I had a gradient, an ecological gradient, to test this hypothesis, whether the morphology was associated with where the snails were found. And so this is the species I used. It's Nesiotis riboschi. And this time I controlled for the fact that these populations are not independent from one another using microsats markers. OK, so here's our expectation. Here, the shell shape going from really skinny to something that's more sphere-like. And here we have island elevation, which is a proxy for how arid the conditions are. And so here's what we found interspecifically. So I was actually a bit frustrated when I saw that, because there's a lot of noise in the data. I mean, it's significant once you control for, so this is the stats once controlled for the phylogeny. But there was a lot of noise, and I was a bit kind of disappointed in the results. But now that I'm coming back to this data set, I'm actually really excited about this, because this means that there's more to it than just the temperature, the thermoregulation, or the water loss regulation across the group. And so I'm now interested in understanding what else is driving the diversity and shape in this group. And I don't have time to talk about that much, so, but you can ask me questions later if you're interested. OK, now intraspecifically, so we're using that single species. And I was really happy to see that the data also fit the pattern where we have like a more elongated shapes at lower elevation. So this is all correlational, and but it still is some indication that there's a relationship between ecology and morphology in that group. So the next step, so this is work that my student Yannick has started at U of I with a collaborator, Jamie Voiles at UN Reno. And now we're really interested to understand what are the metabolic consequences of this shape variation. So we find the slender shape, the snails with the slender shape at lower elevation where it's dry. And so that means that these, presumably these snails are trying to minimize the water loss, which in turn probably has consequences on their metabolic rate and oxygen consumption. And so here I'm showing you is this big data set that we've started assembling for different species. And we're just getting into this. And it's just to show you that we're basically starting to investigate what is the role of this shape variation and metabolic rate. And I don't have like specific data to present to you, more kind of like having like a quick look at the variation that we've looked at so far in this different species. And what we find is generally these skinny species are indeed found at the lower end of these curves. Okay, so I don't have time to go much more into that. I'd like to move on to another aspect of the variation that we see in these snails. And so now what I'm going to show you is work. Okay, so that's like just to show that physiology can actually influence morphology and morphology back physiology. But what we're going to talk about here now is an aspect that I mentioned in the introduction. So how these snails, how these islands can be used as snapshot in time to look at the diversification process. And so I'm going to talk about this in the context of how morphology might be influenced by the communities, the community assemblages where this snail species are found. I'm actually going to skip that for sake of time and move on to the work that my postdoc has started since he joined my lab. And that is looking at the color variation in these snails. So as I pointed out, there's a lot of variation, mostly in brightness, not so much in color. And so this is my postdoc, Andy Kramer, and so he specialized in understanding color diversification in organisms. So he's done his PhD on salamanders, and when he joined my lab, he started working on the snails. So the first thing that we wanted to do is quantify the color variation across this group of species. And so what we did is use reflectance spectrometry to measure color and brightness. And so what this is in a nutshell is basically you have a spectrometer that can measure the amount of light that is reflected off a given surface. And so that allows you to quantify both the brightness, so the total amount of light reflecting off an object, which would be like the area under this curve, and the coloration of this object, so the type of light that is reflected. So basically these peaks that you might have along this wavelength here. As I mentioned, there's not a lot of color variation in these snails. They're mostly brightness. Actually, I think it's 92% of the variation in reflectance is brightness. So for the rest of this part of the talk, I'm going to talk about brightness and not so much of color. Okay, so, well, actually selection of shell color, I guess. Selection of shell brightness. So when you think about these snails that are out there in the habitat, there's various reasons why they would vary in their color. And when I first started working on this group, going from one island to the other, I couldn't see like a clear pattern. I would find these very bright snails and then these dark ones, sometimes in the same habitat. And so we started thinking a bit more about what could be causing this variation. And so one potential selection pressure on these snails in terms of color would be their predators. So a snail that stands out obviously will be more likely to be found by a predator and therefore selected against. And so there's presumably selection for snails to have color, the color of their shell matching the background habitat where they're found. And so I've done obviously quite a few years now of research on Galapagos. And typically when I work on these snails, what I do is I get to a site, collect snails as fast as I can, pack up my stuff, and keep going. And so I started going with my postdoc, and my postdoc is like taking breaks, you know, taking it easy, and like having a little snack here and there. And actually, it turned out to be pretty beneficial, because by staying behind, he had the time to witness things that I've never noticed before. And so one day he just screamed at me, "Hey, Christine, I got something so super cool on my camera." So I go back, and this is the video that he shows me. Let's see if that works. So actually, I'm going to give you a bit of context here. Whoops, sorry. So instead of like packing the snails, so typically we just take like a couple of specimens, and then we measure a whole bunch, but we return the bunch to their habitat. But instead of returning them like under a rock or under a bark or whatever where he had found them, he just left them there in the open. And yeah, yeah. And so this is what happened within like a couple of minutes. So like this mockingbird shows up and proceeds in like just grabbing these snails. And so what the bird is doing is holding the snail by the aperture, by the opening. And then it just hops over to find a rock and starts knocking the snail on the rock. The animal, the soft tissue of the animal is actually attached at the tip of the snail, right? And so it's knocking this off until it breaks open in two. It drops the big part, like the body whorl, the one that's really big and would be a challenge to swallow. And then what's left is like the little tip of the shell and the animal itself, like the juicy, soft parts. And it's just like going at it like pistachio. It's like one after the other. And actually, it eventually finds like its preferred rock. Like then after this video, we obviously witnessed this behavior over and over again. And so what they do typically, they find their favorite rock and just bring the snail over, knock them over, and just keep doing this. And now we've started noticing in the field that there's these rocks, and then like right by the rock, there's these piles of like snail shells just littering around. And so here it is again. So you can see this is like a close-up of the beak. So it's holding it by the aperture like this. It knocks it until this big part flies away, and it then just collects like the other part with the body and just eats it. So in about 10 years of working on these snails, I had never witnessed any predation events. People had mentioned, oh, maybe like there's some beetles eating them, maybe some birds, maybe some lizards. But now it was clear that these mockingbirds knew what they were doing. This was not like a new behavior that this dude just did. And like so we kept track afterwards of mockingbirds' presence and their behavior in terms of predation. So a bird is a very visual predator, obviously. And so you can imagine that matching your background for a snail is pretty important if you want to avoid becoming a snack. So definitely selection on shell color, but mostly brightness again, because like most of the variation that we're detecting is actually brightness. And so what's the prediction under that scenario? Well, in that case, a snail shell brightness should match as well as possible the background where the snail is sitting. So easy, clean prediction here for the biotic selection. But that's not the end of the story. There's other reason why a snail might want to be a different brightness or color. And one that is very important is thermoregulation. I told you on our little hike, the arid zone is super harsh and hot. There's a lot of solar energy hitting that zone. There's not a lot of cover for these snails. And for snails, overheating is actually a very important concern. And so a snail that's bright, or a snail that has a shell that's bright, would reflect more of that solar energy and potentially be in a better shape than a snail that has a very dark shell. And so this works well for snails that are exposed. And what we're finding is that our snail species are either typically on the ground, hidden like in the litter or under rocks or under bark, or they're exposed on the vegetation. We call them arboreal. So they're on the leaves of trees or on the bark of trees, but exposed on the outside. And so we think that this thermoregulation, so the overheating, will be more important for snails that are actually exposed. The ones that are under cover should probably be darker, to actually maximize the solar energy that they would accumulate, because they're in a more humid habitat that doesn't have a lot of, that is typically much cooler than the exposed habitat. And so basically we have these two contrasts, like we have these arboreal species and these terrestrial species. And in this case, our prediction is that regardless of how bright the background is, the species that are terrestrial should have lower brightness, and the ones that are arboreal should have higher brightness. But if we actually put a bit more biology into this, what we would predict is that the points for the terrestrial one would cluster closer to here, because typically background on the ground is much darker than the background of leaves or like the smooth barks, the bark of trees and things like that. And so what we expect is that the points for the terrestrial species would cluster low on the left corner of this axis, whereas the arboreal species should be up here. Okay, so two predictions depending on what's important, whether it's abiotic versus biotic selection, and it could obviously be a combination of both. And when we think about this combination of both, there's actually clear prediction that we can make. So if you go from one island to the other on Galapagos, the abiotic conditions are pretty uniform. It's dry and hot at lower elevation. It gets moister and cooler at higher elevation. Whether you're like on a young island or an old island, that doesn't change. However, selection for by biotic conditions, so by predators, will change if you go from one island to the other. On the older islands where communities are older, there's a lot of birds there, and birds have had time to interact with snails for a much longer time. Presumably, or we expect that this biotic selection will be more important. Whereas on the young islands, on some we don't even have mockingbirds, or we have very reduced diversity in the bird communities. And so then we still have snails there, but we expect that the biotic selection on those younger islands would be weaker. And so here's what we find. So if we look here at the mean species background brightness, right, and the mean species shell brightness, and each one of these dots is actually a different species, what we find is a positive relationship, and this dotted line here is a one-to-one relationship. So when snails are found on dark background, they tend to be dark, and on lighter background, they tend to be lighter. Now let's start to look at these species more carefully. If we break them down into arboreal and terrestrial, that's what we find. So here the circles are arboreal, and the diamonds are terrestrial. So what we find is that the terrestrial tend to be on the lower end of the y-axis in terms of brightness, and the arboreals tend to be higher up. And so that seemed to indicate that abiotic selection is driving this variation here. So we both have abiotic and biotic selection happening in the same system here. Now what about like where they're found, on which island they're found? So here is the map again. So same map that I'm using that I showed you earlier. So here are older islands, cooler end of the gradient, and the red and the pink are the younger island. And so now it becomes clear that the species that are found on the younger volcanoes are way brighter than their background. So these guys are off in terms of matching their background. And so this seems to support our hypothesis that on these younger islands where birds are not so present, what we find is that abiotic selection is much more important. And so what we think is going on is that at first the snails get there, and then they're released from, obviously they come from an older island. So you have the formation of a new island, then it gets colonized from an older island, And so they are released from this selection, this biotic selection from predators, and are free to assume like a brighter color, which is more optimal for the abiotic selection. But as these communities assemble, you get more predation from avian predators, then you get like this matching with the background color of where these snails are found. So here, just to show you, if I actually remove these all the red dots from the relationship, it's almost a perfect one-to-one relationship between the background and the shell color. So once they are in the community, older communities where there's more predation, really the background matching is almost perfect in terms of brightness. Okay, so basically this tells us that ecology is very important in driving the variation that we see in the phenotypic variation that we see in this group of land snails. And this variation that we see is the result of different axes of selection. And interestingly, these axes are different depending on where you are in the process of community assembly. So one thing I didn't tell you is that, here I have a few slides here again, so we're basically going back to it, trying to explain all this variation, is that actually we're using this very old phylogeny, well, very old, 2006, I think, yeah. So this is outdated, it's based on 2 genes, and so one thing that we're doing right now is updating this phylogeny, including way more species, so that we can complete the picture of the variation in this group. One thing that I'm really excited about is that, well, this is going to sound funny, is that like a lot of these snails are going extinct. That's not the exciting part. So, so a lot of snails are going extinct, and it was challenging, or it's challenging, to actually take into consideration these species in the diversification process. But just recently, we've developed a method using ancient DNA techniques to extract DNA out of the shells themselves. So there's no organic material per se in the shell, it's calcium carbonate. But when the shell actually the snail produces the shell, there's little cells, I guess, of the body of the snail that get trapped in the matrix of the calcium carbonate of the shell, and actually provides a pretty much ideal way of preservation for the DNA. Because now these cells are trapped in there and they're really well protected, even though they're lying underground exposed to really harsh conditions. So one thing that we're working on is to put these extinct species back on the phylogeny. There's about 30 of them. And we're really interested in understanding what is driving this modern extinction in this group. One thing that's neat also is, although these snails are going and are really hard to find alive in the field, we can still tell a lot about their ecology just by knowing where the shells are found. So we know if it's an arid species versus a humid species. We'll be able to place it in the phylogeny. We can measure them in terms of their shape and size, and then start asking questions about whether extinction in this group is random, or if it's driven by a certain aspect of either the morphology, the ecology, or the relatedness of the species in the group. Okay, so I'm going to end with this. I'd like to thank my collaborators on this project, namely postdoc Andy, grad student Yannick that started last year, and Jamie, who has helped a lot with the metabolic rate, which I didn't present much data, but I'm really excited about that component of the work. This work is funded by Nat Geo, NSF, and U of I. And with that, I'll be happy to take any questions that you might have. Yes?
I was kind of wondering about the overall species diversity. Do you see more species on the older islands compared to the newer islands, or is it more a matter of the actual surface land area of the island?
Yeah, this is a very good question. So if you, I've done the kind of classic biogeography study looking at different characteristics of islands as potential driver of species diversity. So if you include island area, if you include habitat diversity on that island, island age, island elevation, and there's another one, isolation. So how isolated they are from one another. Turns out that the most important things, if you consider the entire diversity of species, is how big the island is and how old it is. And how old it is. And so then if you actually split the diversity in terms of like if the species are generated within the island or are coming from other source, like from other islands, then if you're just focusing on the ones that are the result of within island diversification, so presumably diversifying within that island, then the only thing that's important or the most important component is habitat diversity. So habitat is really is a big driver to generate species within the island, and then area and isolation and age to a lesser extent is important in determining how many you'll have total, like the ones that are originated within, but also the ones that are coming from other islands. Mm-hmm. Yes?
What's causing the high extinction of these things?
Yeah, we're trying to figure this out. It's a very good question. I guess to get back a bit to your question, so like if you look at the map, the most diverse or the greatest diversity was on Santa Cruz Island, one of the middle islands. And so that's not the, like particularly necessarily large island, but it's really well connected in space, like of where it is. Like it's really central, so like there's a lot coming from other source there. And it's also the island that has suffered most of the extinction. So there were originally 33 species described only on that island. And now we're down to maybe 13 if we're optimistic. So there's like the bulk of the extinction is happening there. And what's happening on this island is an island where it has the largest human population, not too surprisingly. But even outside, so most of the islands are parked area. But on Santa Cruz, you have a big section that's farming zone. But even outside of that farming zone, we find no snails. Like there's like it's almost like a buffer zone around the farming zone where we don't find any snail diversity. And so what we think is going on is that the farming, where they use a lot of pesticide and herbicide and so on, is creating this zone in the highlands where snails just can't survive. And so you had site there where you could find up to 11 species at one given locality. So now it's like all gone. Yeah. And so we think that's probably due to like the overuse of pesticide in the farming area, in part, yeah. Yes?
Is there any correlation between what bird species prefer what snail species, and then is there concern if those snail species are going extinct? Is there worry about the bird species?
Yeah, I'm so excited about this part. So like we just found out about this whole bird predation thing. I had asked in the past bird biologists that work on Galapagos, like, do you think they eat snails? And like I always get like, no, I don't think so, based on stomach content, and like there was no clear evidence. So we don't actually know. So we know that like the mockingbirds definitely are predators. We've seen them over and over again. Finches don't seem to be very interested in these snails. And the other thing is we're just starting to get more detailed mapping of like where these birds are. And again, like the bird people, they just are, typically they're interested in knowing like, is that a bird species that's found in the arid zone or not? But we're working at a much smaller scale of resolution in terms of like where these species are. And so we actually don't have the information whether like the species that we're losing, if they're actually in zones where birds are really active or not. But this is something that we're starting to get more data as we go in the field and collect more information. Mm-hmm. Yeah. Yes?
So on Isabella, you had the red dots were up close to the top. Mm-hmm. Are there no mockingbirds on Isabella?
Yes, but they're not interested in the snails. Mm-hmm. Yeah, they're not as abundant, and their distribution of the mockingbirds is not as spread as on the older islands. So that's more localized where you find the mockingbirds. And when we actually feed them, the snails, they're not interested. And these mockingbirds are Isabella mockingbirds. So there's not a lot of dispersal among islands. I mean, there is a bit, but like not, I mean, these are isolated on Isabella. So is this because they haven't caught up on like the hype of eating the snails, or like they just don't know what they're missing? Like we don't know, but yeah, they seem to be less interested. Yes?
Yeah, I was just wondering if you thought about, in terms of the drivers of morphological variation, if there's any role of predation there, because like in aquatic snails, I think often they're phenotypically plastic.
Yes.
And also heritable variation.
Yes, yes, definitely.
And operculum size.
Yeah, and we're, again, like this is like a new avenue that we're just starting, but you saw like how it grabs on the aperture. So when I started working on these snails, I always thought that the skinny ones would be at a disadvantage, because to me, like it seems like it would be much easier to break like something that's elongated than something that's round. But now that we're considering this, like it's actually probably easier for a mockingbird to grab on the big aperture of the round ones than trying to grab somehow like on these small and skinny ones. And so again, like we don't know enough about like the predation pressure and like how they actually get to the snails to really start answering that question. But this is definitely something that we're looking into. Yeah, but yeah, I'm sure it has to have like an implication on the shape as well. Yes?
So you painted a really interesting picture of sort of community dynamics.
Yeah.
And extinction is a really big word.
Yep.
And I'm just curious if you've considered alternatives to extinction. Like you've only recently discovered this predation pressure from birds.
Yep.
Are you perhaps looking for the snails in all the places that they could possibly occur? Are you considering alternative refugia that may have been lost because of other species extinctions? And looking at it from a community perspective rather than from just the perspective of the snail.
Yeah, and so I've been talking about extinction, but really what I'm talking about is like population declines. And so some of the species we haven't been able to find alive in the field in over like the last, well, since the 60s basically. And so I haven't labeled anything actually officially as being extinct yet, because I'm on the same page as you. Like these snails don't need a lot of space to have a population surviving, and it's really hard to actually survey these islands. And so of course I haven't looked everywhere. So there could be some species that are surviving, but what we're noticing is that some species were very abundant in some areas because we find tons of shells. And like it's like basically the forest floor is littered with these like little bleach shells. And I've been looking in these particular forests for now like almost 12 years. And I can't find any live ones. And so it's more like a measure, we're more using a measure of population decline actually than extinction per se to look at these aspects. Yeah, does that answer your question? Yeah. Mm-hmm.
I would just caution
the
word extinction if they're not actually extinct.
Yeah, no, I haven't put that label on anyone officially yet, yeah. Mm-hmm. Yeah. Yes?
So I was curious, building off of that point, whether the extraction of snail shells has been successful, and whether you've been able to include those in your phylogeny, and see whether maybe you are picking up lineages that you previously didn't have sampled.
Yeah, definitely. So we've been really successful. So like out of like a first round, I think we had like 36 shells that we tried in this ancient DNA lab. We got 17 mitochondrial sequences back. So it's a pretty high rate for ancient DNA process. And so these we've put like tentatively in the phylogeny. It's like a CO1 phylogeny, so it's not ideal. But our next step is, so for the new, like the updated phylogeny, we're using a RADseq approach. And so what we're going to do is try to identify regions of the genome that are more variable or have like high variation, and then design primers for these shorter sequence fragments and amplify those in the extinct species. Because like the challenge with the extinct species is that the DNA is fragmented and shared. So it's really hard to actually amplify big segments. So with the CO1, what we did is like 200 bases or less, and like just kind of like stack them together. So we're going to use a similar approach, but use the information we'll get from the modern the phylogeny from the modern snails, or the ones that are still alive, to design specific primers to amplify highly variable regions of the genome in the extinct ones. Yeah. Mm-hmm. Yeah. Yes.
Well, people have been going to the Galapagos for 200 years, going back and forth, collecting tortoises, and et cetera. How much accidental carrying of snails from one island to another is likely to have occurred? Do you have any evidence?
Yeah, that's a very good question. I actually don't have any evidence of it. I can totally imagine, though, that this could happen. The only, I guess, like our best way at getting at this is to look at the oldest records that we have from the shell collections. And so the first person who collected shells on Galapagos was even prior to Darwin, 1830. And then like we had, well, obviously Darwin collected a handful. And then the CAS had a big expedition in 1905, 06. And so they have a very nice and almost complete data collection from and representing the distribution of most of the islands. And so at that time, there was not a lot of human activities on these islands. This, like, human activities really picked up like in the 50s. And so I guess like I'm not ruling out that like some shells might have moved from one island to the other via human activity earlier, but my guess it would be that like it would have been minimal before that. But yeah, that's I guess like the best I can provide. I mean, colonization, inter-island colonization, we have estimates using the phylogeny, like how often like we have a shift, and it's pretty rare. It's like one successful colonization every million years or something like that. So it's not like that they move around like really freely, extensively among islands. Yes?
So when you did the brightness analysis of the shells, how did you deal with variation across the spectrum?
You mean like in terms of color?
In terms of color. So there are two things that came to mind. One is that presumably with respect to the predation by birds, what would matter would be those spectral peaks that the birds can see.
Yeah, like the UV, yeah, right. Yeah.
And so, but for other issues like thermal regulation, there might be different parts of the spectrum that might be relevant. So there's these black beetles that look to our eyes black, but when you look at them in infrared, some of those ones that live in very hot, sunshiny areas are actually brilliant white in infrared. So it's almost as though it's not a single axis of brightness. There's like multiple axes possible depending upon what spectral range you look at. How did you explore that?
Yeah, so for the bird, what we did is we used like a visual bird model to actually, and this is the work of my postdoc, the details I'm not super familiar with, but basically you can consider the data as envisioned via like the bird vision system. And then so he analyzed the data in that way. And so presumably when he looks at the background matching, this is what he's analyzing is like what the bird would see. Now for the thermoregulation, that's like, that's something we haven't like done yet. And I'd be really interested in knowing like actually how to do this, because we've used brightness and color, and actually both are very similar in the data. I've shown only the brightness data, but the color data, which explain like roughly, it's less than 10% of the reflectance, is consistent with what we get for the brightness data. But I haven't considered that, like that actually we might want to consider, as you say, like the potential that there's different axes of brightness. Yeah. Is there like any papers on that?
I'm not the guy to I just know the one Beatles story about.
Right. Yeah, it'd be interesting. I'll have to look this up. So my postdoc has studied color mostly in the context of predator-prey interaction, and so that's why like all this modeling of like the vision and all that, like so that's like it's pretty solid, I think, with the data that we have. But the thermoregulation is kind of like a new thing that we just started looking at. So yeah, I'll have to consider that.
One paper, the only paper I know.
Okay. Yeah. Yeah, I'll email you then.
Well, in the interest of time, we should cut it short here. So let's thank Christine again.
Yeah. Thanks.